Protein degradation is similarly highly regulated. Proteins get tagged with ubiquitin to then be transported to the proteasome. This tagging is triggered by degradation signals such as the PST sequence or PES sequence or specific amino acids at the N-terminal end of the protein. Protein structure can also influence stability. It has been shown that proteins with many unstructured regions are particularly unstable. So we are interested in the global relationships between these processes. And luckily, during the last years, several large-scale methods have emerged that help us to study this. So we can measure protein and mRNA stabilities using time course data. We can estimate translation efficiencies using polysomal profiling or ribosome footprinting. We can measure mRNA concentrations using microarrays and RNA-seq. However, if you're interested in studying protein expression regulation, you also want to measure directly the amounts of proteins, and that has been much harder. For some organisms, we have libraries of tagged proteins available, but what we are using in our lab is a method that relies on quantitative mass spectrometry to estimate protein concentrations. We developed our method a few years ago. It's called APEX, and we use this to measure absolute protein concentrations in different cellular systems and to compare that to mRNA concentrations to learn about translation and protein degradation regulation. And one of these data sets is shown on the next slide, where we see for over 2,000 yeast proteins, the mRNA and the protein concentrations plotted. And if there was no translation or degradation regulation, every single data point would lay on a straight line marking the diagonal. However, we see a wide spread of the data. That means there's a lot of degradation regulation going on. An R-square value, in this case of 0.58, implies that about 60% of the variation in protein expression levels can be explained simply based on mRNA expression levels. To our surprise, that number was actually smaller in E. coli. Naively, one would think that it's a simpler organism, while as we hear in the other talks today, it is by far not a simple organism. And in fact, the relationship between protein and mRNA expression levels is slightly, it's less well correlated than we see for yeast. In our lab, we're also interested in human systems, and that is shown on the next plot, where we have measured for about 1,000 human proteins drawn from a sample from the Dow medulloblastoma cell line. It's a brain cancer cell line. We have measured the protein expression levels and the mRNA expression levels, and you see here a wide spread of the data, but you also see a significant positive correlation between the two values. Now, we examine this data set more closely because we're interested in explaining what determines protein expression regulation in humans. Given mRNA levels, as I just mentioned, and given our model, we can explain about 27% of the variation. Now we ask, what explains the rest of it? And to answer this question, we go back to that diagram that I presented in the beginning of my talk, which describes translation and protein degradation regulation. And it turns out that all these features, or most of these features, can be mapped to properties encoded in the mRNA and the protein sequences of the corresponding genes and proteins. So these would be the nucleotide and the amino acid composition, codon usage, the translation initiation site, and so on and so on. And we took each of these features, about 200 in total, and asked how much of the protein concentration variation can you explain, given that we already normalized for what can be explained based on mRNA levels. And the set of features that was by far the strongest one was length. So in the protein expression world, size matters. The shorter coding strands in the shorter 3' UTRs correlated with higher protein expression levels normalized by mRNA. And to rationalize this finding, we went back to literature. It has been observed that secondary structures, the number of secondary structures, in particular in the 5' UTR, influences translation levels. So the longer the sequence, the more structures you can have. It has also been observed for yeast that ribosome density decreases in longer mRNAs. It is also, actually that was mentioned in the previous talk already, it has been shown that long proteins have a harder time to fold correctly. So you can imagine that there's a selection against high expression levels of long proteins. And finally, 2 recent papers have pointed to shortened 3' UTRs for highly expressed proteins in mammalian systems, which has been linked to the decreased presence of binding sites for microRNAs that then would suppress translation. The next set of features that had the strongest correlation with protein expression levels were related to protein stability. So we have an overabundance of PST regions and unstructured regions in those proteins that have low protein per mRNA ratios, if you want. And the third strongest set of features that related to the protein expression normalized by mRNA expression were linked to translation initiation. So these would be secondary structures in the 5' UTR, and also upstream open reading frames that are in the 5' UTR and can hinder the ribosome from translating the protein efficiently. And as an example, I'd like to show you the 5' UTR sequence of an so far uncharacterized transcription factor highlighted in red. We see the upstream open reading frame in there. It's a beautiful start codon with an in-frame stop codon. The ortholog of this protein has been shown to be translationally repressed in frog development. So this is a nice candidate gene where we can now show why translation may have been repressed. So finally, in our analysis, instead of looking at each of these features, or in addition to looking at each of these features one by one, we also wanted to set up a multidimensional model that explains protein expression regulation using all of the contributions of all these features. And that is illustrated here. As a reminder, just using mRNA, we can explain about 27% of protein expression variation in human system. Adding length features, we can explain 46%. Now, adding the 20 most dominant features from our analysis, we can explain 2/3 of the protein expression variation in this human system in human brain cancer cell lines. And interestingly, features related to amino acid properties rank slightly higher than nucleotide features, but how and why that looks like in detail remains to be analyzed in future. So to summarize, we've seen that the relationship between protein and mRNA concentrations varies widely across organisms, but we've been able for the first time to describe protein expression regulation in the human system using real data for 2/3 of the data points in our set. And we're now taking this model to other organisms or to other cell lines to understand translation regulation and protein degradation regulation at these global levels. Finally, I would like to thank my collaborators and co-workers, and thank you for your attention.
Great. Thank you so much, Dr. Vogel. Our final speaker for today is Dr. Mark Welch. Dr. Welch studied for his undergraduate degree at the University of California at Santa Cruz and received his PhD at the University of Colorado at Boulder in 1996, where he also carried out his postdoctoral training. From 1998 to 2002, Dr. Welch worked as a staff scientist at Maxigen in the development and application of directed biomolecule evolution. He then pursued work in the area of rational and combinatorial protein engineering and kinetic analysis at Kozan Biosciences before holding a staff position in the Advanced Research and Technology Group at Applied Biosystems, focusing on engineering of DNA polymerases with a variety of altered properties for new and improved applications. In 2007, Dr. Welch joined DNA 2.0 as Director of Gene Design to develop the company's capabilities in the areas of protein expression and engineering. He has more than 12 years' experience in directed evolution and engineering of nucleic acid and protein biomolecules. Welcome, Dr. Welch.
Thank you, Sean. So our focus is somewhat different from that of the previous two speakers. Rather than the underlying mechanisms that control natural gene expression in cells, we're more concerned with how cells respond to unnatural heterologous expression. So heterologous expression is often a barrier to protein study and manufacture. We want to understand, and often the problems with expression are rooted in the gene sequence itself. We want to understand how to best design genes to circumvent these problems. So traditionally, the way people have approached this is to guess essentially what's important in the cell based on our limited knowledge, and you've heard about our limitations that currently exist. basically guess what the cell prefers, and then come up with schemes, and you can see several different schemes in the literature that approach it this way. Problem is that most of these are mutually exclusive, and several are really poorly validated. Instead of just guessing what the cell prefers, our approach is to simply ask the cell. So the way we approach this is similar in principle to what we heard Dr. Plotkin talk about, except that instead of using random variation, we use design of experiments methods to broadly sample gene design parameters of interest, and then use gene synthesis so that we can sample any region of parameter space that we're interested in, and we can sample a broader range of space with as few genes as possible. And then just test these genes in E. coli and let the data tell us what's going on. So the work I'm going to talk about is where we've done this for 2 different proteins in E. coli, a DNA polymerase and a particular single-chain antibody. And most of the details of this data are published recently. So this just graphically represents the diversity in our antibody set. Each column in this matrix is a discrete gene in the variant set. Rows indicate sequence features, such as codon frequencies and other features that have been associated with protein expression by others. And the colors indicate whether the features are accentuated in the gene or diminished. What we see when we diversify genes this way, and we see this for both this set and the polymerase set, is a wide range of expression levels from below our detection limit and over a range of 2 orders of magnitude up to 30% of the total cell mass, a very high overexpression. So we're sampling variables that are critical for expression, clearly. The question is, what are these? Similar to what we saw with Dr. Plotkin's study, we don't see a correlation with the codon adaptation index. Now, we're sampling a range of indices of this index that's more relevant to gene optimization strategies, a higher range than what you see in the GFP study. Yet still, from either of our data sets, we don't see a correlation with CAI. In contrast to what we saw with the GFP study, we also don't see a correlation with 5' mRNA structure strength. And what's assessed here is the window that was shown for GFP to correlate best with expression. We've also looked at windows upstream and downstream of this, and we haven't found any correlations in structure to date. But this isn't necessarily contradictory to what we saw with the GFP study, because the sampling range of structure strengths that we're looking at here in our library are far weaker than what was observed to have a significant effect in the GFP library. We think this is simply a product of the different amino acid sequences at the end termini of these proteins versus GFP. If you look at the series of variants indicated by the red arrow, we see a number of, these are all variants that use the same coding usage for the same sequence for the first 15 codons of the gene. Yet we see that their expression levels span over the entire range of our data set. So clearly, there's a lot of variability that's not explained by just the terminal coding sequence. So we wondered whether the predominant effects were global in nature, distributed, or local, like deleterious motifs. And one way we approach this is to make gene chimeras from genes in our data set. So in this example, we took 2 genes, a weak expressing and a strong expressing polymerase gene, and split them into 3 parts, and then made all the combinations of the segments and looked at what expression pattern we see. And what we see is, in this set, is a distribution that all the segments are important. If you make single replacements of the good gene in the bad gene, you see mild or no significant improvement. So you can't localize the effects to single motifs in a particular region. If you make double replacements, we see some boost in expression for all the double replacements, but we don't get as high expression as the best parent. So the effects seem to be distributed throughout the gene. We can't confine them to the 5', the middle, or the 3' ends. So this led us to reconsider whether global parameters are at play, such as codon usage. So we've heard in both our talks of mine and Dr. Plotkin's that there's no correlation of expression with the particular direction in codon bias represented by CAI, by the codon adaptation index. But we asked whether there's any direction in codon bias in our data set that correlates